Hello, my name is Kusha, and today I'm going to talk to you about my summer project, calcium dynamics and myelination. Last March, a lot of us found ourselves with more time on our hands than we were used to, and different people I've spoken to decided to use this time in different ways. For example, my parents decided they wanted to learn a new language, I have friends who tried their hands at new instruments, and personally, I wanted to learn how to ride a skateboard. But have you ever wondered, what changes occur in your brain when you learn a new skill like this? If you were to take a snapshot of your brain before and after you learn the skill, what would look different? The particular research question I was interested in answering was, how does the activity of a neuron, a cell that transmits information from place to place in the body, impact its signal conduction? How well and how quickly it's able to send that information? As a bit of background, neurons have these long arms that transmit electrical signals from place to place. Now, it's important that these signals are sent both efficiently and quickly. So in the central nervous system, there are cells called oligodendrocytes that enwrap these neurons in a fatty, insulating material called myelin. Myelin works a lot like rubber on a copper wire. If you send electricity through a copper wire with no rubber, a lot of it is lost, and the signal fizzles out and usually travels pretty slowly. But as soon as you enwrap the copper wire, the signal is able to be transmitted much more quickly because less energy is lost to the outside environment. There's still a number of things we don't understand about oligodendrocytes. For example, how do they decide which neurons to enwrap? And what determines how much myelin there is enwrapping a neuron? In order to answer these questions, these experiments looked at calcium, an important signaling molecule implicated in things like bone solidification and muscle contraction and myelin sheath growth. They found that calcium waves in the oligodendrocytes were strong indicators of whether the myelin sheath grew or contracted. They found that higher frequency calcium waves usually led to myelin sheath growth. More importantly, they saw that myelin sheaths that enwrapped axons that were more active had faster calcium oscillations. This creates the framework for activity-dependent myelin plasticity. This is a feedback system where a neuron, if it's more active, has more myelin sheath growth. It's then able to send signals much more quickly, and then it's able to become more active. Experiments have actually shown when people learn a new language, the gross myelin volume in their brain actually increases. If you were a biologist looking at one of these cells, you might create a movie like this. This movie tracks the movement of calcium in space and in time in the complicated branching structure of oligodendrocytes. You might think, how could somebody understand something as complicated as this? This is where mathematical modeling comes in. Mathematical modeling takes a mechanism that's not fully understood and experimental data describing the system, and it makes a guess at how it might work. That guess is then converted into math. The math is used to run a simulation, and eventually the simulation is used to make predictions and suggest experiments to test a theory about how the mechanism works. So at the beginning of the summer, I was fortunate enough to receive some data of calcium in the arms of oligodendrocytes. Then, using what we already understood about the system and characteristics of the data, we were able to represent the mechanism as a model. Now, this is usually done using differential equations. That means equations that describe how things change instead of their absolute quantities. This can be really useful in biology because it's really hard for me to tell you exactly how much calcium there is inside a cell at a given point, but I can tell you how that calcium changes because I know what receptors there are regulating the flux, and I know what forces drive the movement of calcium. Using this model to run a simulation, you might get something like this that predicts the calcium waves in oligodendrocytes. I want to add that our work isn't done. There's still a number of characteristics that we haven't yet captured, but what's interesting is we can take a word model, a general idea of how things work, and translate it to data like this. The hope is if we better understand calcium transients in these oligodendrocytes, we can understand how activity-dependent myelin plasticity works. I want to thank you for listening to my presentation. I want to thank MiCM and Dr. Kadra for supporting me over the summer. And if you're interested in my work or if you have any questions or comments, please don't hesitate to email me. Thank you.